Good afternoon, ladies and gentlemen. I'd like to thank the organising committee for allowing me to present my research on the genetics of cancer cachexia. I'm going to tell you today about an exciting new novel biomarker that may be able to predetermine patients that are liable to suffer from cancer cachexia and how they are genetically may be predetermined to go on to develop cancer cachexia. Secondly, I'm also going to tell you about a phenotypic correlate in human skeletal muscle, which up until now has been able to validate a particularly arbitrary diagnosis that is cancer cachexia. So cancer cachexia is a devastating syndrome, and it represents a major unmet clinical need characterized by a number of symptoms that ultimately lead to reduced survival, impaired tolerance of surgery, radiotherapy, and chemotherapy. It's estimated that 80% of patients with terminal cancer die as a result of cancer cachexia. In the post-human genome era, one of the major goals of biology is to successfully link the gap between genotype and phenotype. It all starts with a patient, and it's thought an interaction of a variety of mechanisms, including host genetics and tumor phenotype, lead to the cachexia phenotype. In terms of the tumor phenotype, stage, type, and differentiation lead to the cachexia phenotype, but it's thought that host genetics play a role in the development of complex multifactorial diseases such as cachexia. And these are alterations in the base level of DNA, termed single nucleotide polymorphisms, which leads to alterations in how a gene functions. The aim of this project was to characterize any association between single nucleotide polymorphisms and the predisposition to develop cancer cachexia. All phenotype-genotype association studies rely on accuracy of phenotyping, and it's imperative to pin down a phenotype. Up until now, these arbitrary diagnosis of cancer cachexia has been based on weight loss and low skeletal muscle by a group of experts with no tissue validation of these definitions. Up until now, I was able to take 40 patients and biopsy their rectus abdominis muscle and stain for myosin heavy chain. If you look at the patient on the left, this is a control patient with no weight loss, and pay attention to the fiber diameter. If you move to a patient with cachexia defined as a gross degree of weight loss greater than 10%, you notice no real difference. However, if you add a level of skeletal muscle index into the definition, you see a dramatic reduction in the fiber cross-sectional area of their muscle. I was able to determine skeletal muscle index based on pre-operative staging CT scans. These are all 6 different patients. If you take the top row, they all have identical skeletal muscle indexes, but hugely varying degrees of BMI. Whereas if you take a routine measurement that we're all very used to, BMI of relatively healthy, 29, you can see this patient down here has a skeletal muscle of 33.7. So if they're sitting in the clinic, they have a dangerously low chance of not making it through surgery, as has been shown to be a powerful prognostic indicator. So having accurately characterized the phenotype, I moved on to the genotype, and the case for a genetic predisposition to cancer cachexia is further strengthened by known mechanisms in inflammation, muscle tissue biolysis, fat tissue biology, central homeostasis mechanisms, appetite, and others. All these have known roles in cachexia and wasting diseases. I identified 154 genes in a systematic review and found 281 functional SNPs that affect protein function. I recruited 1,300 patients, of which I obtained whole blood from and obtained genomic DNA through this. 102 were recruited for a transcriptomic analysis. I adjusted with the following co-founders and performed a power calculation to achieve a power of 80%. The genotyping was performed with sequencing norm IPLEX Gold platform to enable radio detection of MALDI-TOF mass spectrometry. Moving on to the microarray, use the AlGenet platform. And these are the demographics. I split into a validation and a discovery cohort and a combined cohort. As you can see, we used the pure weight-losing cancers, such as upper GI cancers, esophageal or gastric, and lung. There's no statistical difference in any of the variables. So moving on to the results. We were able to confirm from a previous stage 1 analysis via a stage 2 analysis in a validation cohort that the SNP RS6136 in the SELP gene, which encodes for p-selectin, which changes the amino acid sequence. It's a non-synonymous change in the DNA from a threonine to a proline, and this was significant in all degrees of weight loss, whether it be 5%, 10%, or 15%. Moving on to a discovery cohort for new SNPs based on the new diagnosis and also varying degrees of weight loss. I managed to find a number of SNPs that were statistically significant after multiple testing in the following genes. And the ones highlighted were of particular significance. Adding in a level of skeletal muscle index, we were able to identify these genes and the SNPs in these genes. And again, one of particular interest. And why these genes and SNPs are of particular interest moves on to the well-known ubiquitin proteasome pathway and TGF-beta signaling that acts that is well known in degrading muscle. Myostatin and activin bind to the ActR2B receptor, which leads to downstream signaling of SMAD2 and quiescence of satellite muscle cells. Activates transcription factors, which translocate to the nucleus and enhance transcription of the E3 ubiquitin ligases, estrogen-1 and MERF1. And these are well known to degrade skeletal muscle, as I've been able to show before. All these proteins were, transcriptomic analysis was performed. I don't have time to show the slide, but they were statistically significant after multiple testing in terms of representation. So the SNPs we have found with 1 in ActR2B and 3 in the FOXO1 and 3 transcription factors. So in conclusion, ladies and gentlemen, I'd like to say for the first time that we've been able to validate in a very large cohort a potential new biomarker which will be able to screen for patients that are predisposed to developing Cachexia, and in conjunction with pharmaceutical company Novartis, we're able to start a screening program. Secondly, we've been able to look at skeletal muscle and show a novel phenotypic correlate which validates for the first time the diagnosis of Cachexia, and this can be used for endpoints in complex interventional trials. And thirdly, I've identified a number of SNPs that are new in a discovery cohort which will need validation in future. So I'd like to give the following thanks to making this possible and invite any questions.